A quick review today, kiddos, of how to do a Punnett square. Remember, Punnett squares are tools that show us the probability of passing on different genes from parents to offspring. So let's read the problem, break it down, figure out what the parents' genotypes are, and then what they pass on to their offspring. So first it says cross a heterozygous, keyword there, heterozygous means different, a heterozygous mouse with a white mouse. So it says gray is dominant over white. So gray is dominant over white. Use letter G. Well, if we have heterozygous, heterozygous means different. So we have a big G and a little g for our alleles. Now I could put that across the side, but I just put it across the top. One letter above each box. So big G, little g, that would be one parent we have, heterozygous mouse. Our other parent, it says with a white mouse, and I don't give you any other information, I just say white mouse. So how are you supposed to be able to figure that out? Well, I also say gray is dominant over white. Remember, dominant traits always show up if they're there. Recessive traits only show up if there's no dominant or if there's only recessive traits there. So if gray is dominant, we know capital G represents dominant, and we know lowercase g represents recessive alleles. So we know this right here would give us a gray phenotype because the capital G is dominant, gray is dominant. If we had this, two capital G's, that would also give us a gray phenotype. The only way to have a white mouse is to have two recessive alleles or two lowercase g's for our genotype, and then that would mean the phenotype or the physical appearance is going to be white. So now we have our genotypes. We have heterozygous genotype and we have a homozygous recessive genotype. So here's our heterozygous mouse. It has to be gray, right? Because gray is dominant. Anytime you have one dominant allele, that's what shows up. And then this over here, this parent over here, is our white mouse because it's got two recessive alleles. So our Punnett square, we just drop down an allele, bring one over. So I got big G, little g there. On this side, little g and a little g. Over here, drop down the big G, bring over the little g. And then we have drop down our little g, bring over our little g, and that would be the probability we have for our students, for our mice. Now, we got to kind of break this down. So these are the different genotypes. Remember, genetic makeup. Those are our genotypes. So it says what percent of the offspring, the babies, are homozygous. Homo means same. So of these offspring, how many are homozygous? Well, they're the same. They're the same. So 2 out of 4. These are different over here. So we got 2 are homozygous out of 4. That would be 50% of the babies or offspring are homozygous. So that means 50% is heterozygous. Now, what is the phenotype of the offspring? Remember, phenotype is physical appearance. Well, let's look. If I have 2 lowercase g's, remember lowercase g, that's recessive, or that's going to be the white. So we're going to have 50% of the babies will be white colored. Now here I have one capital G and one lowercase g, one dominant allele, one recessive allele. Anytime we have a dominant allele, the dominant shows up. So these are both heterozygous, one dominant, one recessive. The dominant allele shows up, so I will also have then 50% gray because gray is the dominant color in this problem. So my ratio would be 1 to 1, or 50% white to 50% gray.